So it's my honor to introduce the next speaker. Dr. Franklin Carrero-Martinez is the Senior Director of the Science and Technology for Sustainability program at the National Academies of Sciences, Engineering, and Medicine. He earned his PhD in Cell and Developmental Neurobiology and a certificate in Business Administration here at the University of Illinois. Over the course of his career, he has served as researcher and educator, science administrator, and science diplomat in government, academia, and various NGOs. While at the University of Puerto Rico, he established a strong research program aimed at understanding the brain self-organization principles and how to use that knowledge to manipulate brain connectivity. In 2012, he transitioned to government service as an American Association for the Advancement of Science Roger Revelle Fellow in Global Stewardship. Dr. Carrero-Martinez has held many positions in government, and as of 2018, has held his current position at the National Academies of Science, Engineering, and Medicine.
Thank you very much. Can you hear me well?
We can.
Yes.
Okay, thank you very much. So for me, it's a great honor. After I graduated from Illinois in 2005, I have not been back, and I was really hoping to be back, but COVID had other plans. So I'm glad to be here. I'm glad to see all of you this morning. I was quietly listening to all of the talks. Thank you for giving me the luxury of listening to all of these talks. It's been a while since I've been able to attend a scientific meeting. I want to start with a little bit of what I've been doing since I left Illinois, a little bit of the research that I did while I was there as a trainee of this program in its early days. And then I'm going to move to what I was doing at the University of Puerto Rico, where I was a faculty member and I had a research lab. And then the transition that I did to government service and what I'm doing now. So I'd like to start with this quote. A lot of my colleagues and other former students think that I have a interesting life. Sometimes that may be the case. Sometimes it's very stressful with some of these things that are described here being true, but it's part of the journey. So my talk today is called Understanding the Brain, a journey from the lab to the halls of the UN. And I will share how I went to the lab into literally the UN. So after I left Illinois, I was a professor at the University of Puerto Rico in Mayagüez. I was an adjunct faculty at the University of Puerto Rico Medical Sciences campus. I was a visiting scholar at Duke University, and then I did a mini sabbatical at the MIT Picower Brain Institute. Then I transitioned to government. I spent time at the Department of State, at the National Science Foundation, again at the Department of State. I was a science attaché at the embassy, the US Embassy in Mexico City. Now I'm a part of the National Academy of Sciences, and I also have an honorary professorship at the University College London. So during my time at UIUC, I worked in the lab of Akira Chiba. I'm still in touch with him. We have been in touch discussing a few ideas and picking up some of the research that his lab had continued to do based on my research. So I serve quietly in the background sometimes as a consultant, and I have the opportunity to geek out and be a little bit of a nerd on some of the things that I was doing during my PhD.
Excuse me, I'm sorry to interrupt, Franklin. Would it be possible to change the view of your slides to, we see all of the slides at once?
Let me see how I can do that.
There, that's much better. Thank you.
Okay, absolutely, thank you. So I wanted to look at and understand the brain. That's one of the reasons why I joined the program in Illinois. And I was very interested in figuring out how to provide tools and develop tools to manipulate the brain. Of course, in humans, that's not possible, and certainly was not possible 15 years ago. So we started with small brains, and we focused on fruit flies, the fruit fly brain, specifically the neuromuscular junction. did a lot of imaging, a lot of characterization of neurons and muscle behaviors, and looking at the different molecular components that manipulate, that allow the brain to self-organize. Had the opportunity to learn a lot of techniques, visualization techniques, imaging techniques, that oddly enough have become very helpful as I have different roles in government. This is one of my favorite movies of the time. I'm sure that the resolution and things have changed significantly from what we were doing 15 years ago. These were some probes, bioprobes that we developed, genetically encoded probes that we developed in the lab that allowed us to visualize the neuromuscular junction formation in vivo as the organism was developing. Part of what I was doing in particular was looking at, this is a brain dissection of the fruit fly expressing GFP, which I think with all of the coffees that I had since, I've lost the ability to do. But it was very interesting at the time to observe very fine processes that the muscles put out. The lab dubbed them at the time myopodias, as opposed to philopodias coming from the neurons. And we were looking in particular, specifically these neurons and these muscles in this part of the brain. And these are the myopodias. Unfortunately, I don't have the movies that would accompany this, but these processes were very dynamic. And we, at some point, described that they had adhesion molecules at the tip of those. In a way, it facilitated the interaction between the neuron and the muscle as the neuron was approaching. And this is how we were visualizing using various probes at GFP, RFP at the time, encoded in both neurons and muscle to visualize the process of synaptogenesis. It reminds me to that quote from Ramon y Cajal when he described the protoplasmic kisses that form the ectasis of a neuromuscular junction formation. So this is one of those very cool experiments that I was able to do at the time, and look at the behaviors of the different cells to get to the point to form a functioning synapse. We did a lot of live imaging in multi-dimension, characterized the myopodial cluster, identified underlying adhesion molecules, scaffolding proteins, and other things that put together the myopodia cluster and the signaling cascades that eventually led to the formation of a fully functioning synapse in the developing embryo. After that, I had the opportunity to visit the lab of a researcher in Japan. I did a short internship there at the Institute of Genetics in Mishima. It was a very interesting experience. I learned a little bit of Japanese, which I have since lost because I'm no longer using Japanese, but it was a very interesting experience that enhanced my connections to the international scientific community. This is a picture of the lab after my thesis defense in 2005. I kept in touch with most of the colleagues in the picture throughout the years. And then I moved to the University of Puerto Rico, where I established my lab. I continued part of the research that I started with Akira, and started looking at the molecular components on the postsynaptic side that would allow for the neurons to remain connected. We did a lot of genetic manipulations and a lot of different tools that we were able to develop that led to a couple invention disclosures and a couple of other very interesting things. Particularly at very high resolutions, we were looking at vesicle transports on the postsynaptic side as a result of muscles, of the neuron, I'm sorry. And this is one of my students, which graduated from my lab as an undergrad. I had around 25 undergrads, and a few master students that went on to different schools here in the US mainline to complete their PhDs. So from that research, I started developing additional collaborations with colleagues in other departments and other schools. And we started developing nanomaterials with the intention to manipulate the nervous system. So using the tools that we had developed, what I wanted to do was develop ways that we could reconnect neurons that had become disconnected for a variety of reasons. That could have been trauma, genetics, or any other kind of issue that might have come up, and we would try to reconnect. That was the basis, the underlying idea of that project. We had funding from the Department of Defense to do this at the time. And we started developing some tools, very cool tools to do that. Here's another movie when we were characterizing at higher resolutions at the time, the neuromuscular system, in order to facilitate tagging of the nanomaterials. And I'll share with you some what those look like. We also started characterizing a few neuromuscular junctions at very high resolution with the idea that we would have a catalog to establish the normal baseline, and then from there see what we could do after manipulation. That led to a series of very interesting experiments and collaborations. I still stay in touch with some of those researchers, even though I left the lab about 10 years ago. But one of them was with a group of chemical engineers that helped us develop very specific nanomaterials that would allow us to paint the neurons and use that to engage them magnetically. That also led to solving a few very interesting problems that we encountered when trying to engage things without touching the embryo magnetically. So we developed a collaboration with a physicist and developed this tool that we mounted in the microscope to engage the nanoparticles magnetically. It led to a few interesting things that at the time escaped my brain in terms of the complexity and what they were doing, but we were able to, at least from the biologist in me, I was satisfied that we were able to engage the nanomaterials in vivo with this magnetized tip, engage them magnetically and change the direction. And by doing that, then create a force that would literally move the axon. That was like a 2-year project just to get to that point, but we were able to not only image, but at the same time engage the cells magnetically. To do that, we had to develop additionally protocols for painting the neurons with the nanomaterials. It was a little difficult to pull off, but I had very skilled students. I guess they spent a lot of time playing video games, and they thought all of the manipulators and actuators in the lab were similar to those. So they, I guess, developed the fine motor skills required to do that. And they were able to paint the neurons. So going back to this diagram, this is an example of how the painting of the nanomaterials that were tagged with specific antibodies to neurons, we were able to do that. So we were super happy. We started engaging the cells magnetically. This is, let me jump to this. So this is how it looked. So we had specific tagging using proteins that were sitting at the surface of the neurons. And this is how it looks. We did local painting using microtransfer. But unfortunately, one of the things that we started discovering was that the embryos would die. This is how it looked. So this was the purpose of the experiment. So we had this neuron, which I showed you before. It has the neurophilopodia. The muscle also puts out the myopodial clusters. And the idea was to engage the neuron magnetically and move it to a different location. We were able to do that successfully, but the embryos were dying because of toxicity from the nanoparticles. So of course you can move anything, but it doesn't serve any purpose if it's dead. Regardless, the main idea at the time was trying to figure out at the molecular level which cell is driving the communication. The question still remains, is it the muscle telling the neuron that this is where I'm ready, you need to come here, meet me here, and make the connection, the synapse here? Or is it the neuron telling the muscle, I'm going this way, so please get ready? And that's what these processes that I showed you earlier are doing. Short answer is that we were not able to answer that question and still remains an open question. So if any of you have any cue or clues, please let me know, because I'm still, even 10 years after, I'm still interested in finding an answer. One of the other things that we were doing simultaneously was using single cell laser axotomy. And you see this movie here. We have the myopodial cluster connected with the neuron connected. That bright light was the laser. And we cut the muscle and came in, painted the neuron, and we're literally pulling the neuron back to the muscle. In some cases, very early on, we saw the beginning of a reattachment, but again, the embryos die because of the toxicity of the nanoparticles. This was the underlying hypothesis that we were exploring, that the interactions between the two cells would trigger adhesion molecules at both the neuronal surface and the muscle surface, and that would then recruit other molecular components. So the idea and the hope was that eventually we would be able to provide to facilitate a functional neuromuscular junction after attempting to reconnect. This is how the embryos look like after microinjection. So that led to a long process, and I'm not going to bore you with all of the details, to figure out nanomaterials that were compatible. We also found out that the nanoparticles would accumulate in certain tissues with preference, and eventually we developed a few nanomaterials that were able to not to accumulate inside the body, would not cross membranes, and would remain viable as for tagging and as a fluorophore that we could use to paint the neurons. But unfortunately, around that time, students engaged in a protest at the university. It led to the university being closed for a couple of months. I lost the research. The university lost accreditation. I was put on probation from receiving federal funds. So at that point, that summer, I went to MIT, where one of the faculty members told me that maybe I should look at doing something else. And I told him that I did not know what else to do, because I had trained to do the job that I was doing. And he suggested that I look another way. And he suggested that I looked at science policy. I did not know what that was. So I started the process of exploring, and led me to a journey of discovering what my next interest and my next career move would look like. But most importantly, a reflection of what I had done in the past couple of years at the university. So in that time at the university, I had a few invention disclosures. I was lucky enough to have very good students in my lab. I also developed a new line of research within my lab on STEM education looking at what are those factors that encourage underrepresented students to choose and persist careers in science. I had funding from NSF, Howard Hughes, and other institutions, the Department of Defense, to look at that and provide support for those students. I taught thousands of students in my undergrad classes and graduate classes. I can tell you that I was not the favorite professor, but I had fun at the time. And then what I used was the international connections and experience that I had before to do that career transition. So I transitioned to Washington. I came via a fellowship from the AAAS, that's the American Association for the Advancement of Science. I was named the Roger Revelle Fellow in Global Stewardship. And I had a dual appointment, and a 50-50 appointment never works at 50-50. So I had like a job of 150% effort. The idea of that fellowship was to provide a unique opportunity for scientists to address global stewardship issues. And that's what I started doing, bridging the gap between what I was doing before and the new role. So at the Department of State, I was at the Office of the Science and Technology Advisor to the Secretary of State. And simultaneously, I was at the National Academy of Sciences in the division called Development, Security, and Cooperation, which no longer exists. And a lot of people at the time asked me, so how do you go from a neuroscientist to now working on all of these issues? And I always found this analogy very interesting. And this is what I want to call your attention to, in case, just so that you are mindful and thoughtful of what you're doing now and can see and understand the broader implications for your immediate future or for the longer term. So one of those things that I told people is like, so here's a city and it has the same level of complexity as a neuron. And when you look at it from different perspectives, yes, we can argue about the details, but from the cytoskeleton to road networks, from vesicles moving to buses and cars moving around, to borders and very distinct membranes, you can make a lot of parallelisms and a lot of connections between one and the other. And I would encourage you to consider being mindful and aware of what those are as you communicate your science and your research to others that may not necessarily be in your field. So what I ended up doing was, and what I'm doing now, it's focusing on science diplomacy. I will share with you what science diplomacy is and the different dimensions of science diplomacy, which is what I'm doing these days. So the interaction or the intersection between policy priorities for the US government and foreign governments, that circle at the bottom here, that's what diplomacy is. When you take a look at this two groups represented here, the scientific community here in the US, scientific community overseas, where the two cones intersect, that's what we call science collaboration. And we have, of course, the interaction on the US side. We have the US scientific community talking to the government. That's scientific advice, advocacy, lobbying in some cases. And you have the same thing going on overseas between the different scientific communities and their governments. Where those two, the diplomacy and the collaboration circles intersect, that little spot is what science diplomacy is. So some people try to define science diplomacy as this super big thing, but in fact, it's not. If we zoom in even more to look at what science diplomacy looks like, we can further divide it in two. So we have diplomats that do have an interest in science. Those are representatives of the respective governments. I had the opportunity to serve in that role when I was at the Department of State and as a science attaché in the embassy in Mexico City. or scientists that work in diplomacy, which is what I'm doing now at the National Academy of Sciences and what I was doing when I was a fellow at the AAAS. So what is science diplomacy? Science diplomacy is the use of scientific collaborations among nations to address common problems and to build constructive international partnerships and understanding. These are the kinds of science diplomacy that exist. We have science in diplomacy, which are scientists providing advice to inform and support foreign policy objectives. We have diplomacy in science. Those are diplomats facilitating international collaboration. We have science for diplomacy, when the scientific cooperation improves international relationships. And we have the science of science diplomacy, which is an emerging field looking at how the scientific community and the diplomats to come together to advance science diplomacy. So that is the conduct or the study of science diplomacy. There are different kinds of diplomacy. The one that the government conducts, we call it track one. The one that I am currently conducting on behalf of the government is called track 1.5. That's when we, organizations such as like the Academy of Sciences, where I am now, facilitates conversations on behalf of the government. Or I should say, not on behalf of the government, but in coordination with the government. That's the track 1.5. Track 2 is when organizations do things to advance the interests of the host government, but not necessarily in coordination with that government. And then track 3 is when organizations do whatever they can to go against established policies of the host government. At the Academies, we focused on 1.5 and 2. We don't do track 3 diplomacy. When I was at the Department of State and NSF, I had the opportunity to participate early on the BRAIN initiative that the Obama White House put together. It's a partnership. I think there was an article that came out recently in the last couple of days doing a recap of what the BRAIN initiative had done as we get close to the 10-year mark of the BRAIN initiative. I had the opportunity from the NSF side to implement and develop some grants, not only for basic research, but also for facilitating STEM education. And when I moved to the Department of State, I coordinated at the UN a few meetings and high-level interventions that brought the BRAIN initiative to the international stage. It was a tremendous lift with a lot of government partners. Here, there's a picture of the previous NSF director, Franz Cordova, delivering remarks to encourage countries to engage with the BRAIN initiative in the US and start coordinating their projects. There was a lot of overlap and a lot of gaps between the different BRAIN projects. And this is something that we did at the Rockefeller University and the UN simultaneously. So we had a 2-track event, one for the scientific community and one for diplomats. Here I am with the Swedish science minister and the science attaché, and Nobel laureate Torsten Wiesel, who was very kind and instrumental in putting some of these meetings together. This is the UN building in New York, and I'm going there tomorrow for engagement at the UN General Assembly. Here it's a picture of the NIH director, current NIH director, Dr. Collins, and Dr. Cordova, the previous director of NSF, and the science advisor to the Secretary of State. Later, I'll share the story of how I was able to smuggle them into the UN with passes from other countries because the US government couldn't facilitate those for them. And here in this other picture, the Obama science advisor with the NSF director and NIH director. Here, I always like this picture because Dr. Wiesel was, this is the UN ambassador from Argentina. And Dr. Wiesel was essentially telling him, you look disheveled, you should organize your name tag and put it together, you're an ambassador, which was always like very interesting. And here, a picture of Secret Service that was looking at our meetings from behind the scenes. I want to share a small video that some of the partners put together for this engagement.
When you build a building, you have a set of blueprints, and an architect and engineers sit down and design a communication system, plumbing, air conditioning, all of that. In the brain, we're actually trying to reverse engineer that away. We actually don't have the blueprint.
Possibly the most complex entity in the entire universe. There's microarchitecture, nanoarchitecture, macroarchitecture, and then there's the function of the brain, which we observe in everything we do when we interact.
As humans, we have this intellectual curiosity about what makes us different. It's all related to our brain circuits. Getting the tools to allow us to see circuit activity in action and then apply it to problems that no one's been able to solve before would be a tremendous advance.
We all have a brain and we want to understand that brain better. Get the scientists from different communities, different cultures, different emphases together. They'll start forming their collaborations organically. To do that type of sharing, you don't need a big bureaucracy. You don't need a lot of complexity. You just need to talk to each other.
No one person or even small number of people have the expertise in terms of anatomy, physiology, computation, theory, mathematics. We can all act synergistically to come to a better model of what's going on. We don't know how to cure depression. We don't know how to treat Alzheimer's. We don't know how to deal with post-traumatic stress disorder. When you have diversity in a group, you would take in the advice, experience, expertise from others. And that, I think, at the end will end up enriching whatever you're trying to build.
Brain. When you build a building.
So you probably know what happened with the Brain Initiative. We had a change in administration, and then we had to move to work with other partners to park the Brain Initiative outside of government. And the Brain Initiative continues, I believe, with the Kavli Foundation and other organizations that continue to move that initiative forward. After that, I moved to international negotiations at the multilateral level. This is how a negotiation room at the UN looks like. This is in the introductory slide. I mentioned the boredom to death. This is what it is. Sometimes you're fighting for a comma, a period. Is it should, is it could, is it would? All of that really matters when documents that frame or form the basis for our scientific collaborations. You may not necessarily be thinking about it, but there is a framework if you're moving samples from the US to another country or the other way around. There are rules, there are regulations, and most of them come from negotiations at the UN. This is a group of us at the UN. Some of those negotiations are very difficult, not only for the meaning, but also advancing US interest and trying to reach consensus on the interest of 190 plus countries. It's sometimes not easy, but that's part of the job. The meetings were long, but you were trying to provide the best way that we can form basis for global collaboration. This is an example of when I was in Mexico. This is the late Mario Molina, Nobel laureate in chemistry. His research led to the discovery of the fact that fluorochlorocarbons are having an impact on the ozone layer. Eventually, he turned to advocacy role, which led to the Montreal Protocol that banned CFCs. I did a lot of engagements there in public, that with the different parts of the embassy, had the opportunity to teach a couple of classes and meet a lot of people, including organizing the first international visit that Dr. Cordova had to Mexico with then Secretary of State John Kerry. I also had other engagements in other parts of the world. This is the Pakistan portfolio that I had. I've been fortunate to not only work with great colleagues there, this is the picture of the ambassador, but also they took me hiking. And I'm not sure how many people can say they went hiking in Islamabad, but I had a good opportunity to see other parts of the country. And then came back a couple of times. I've been in Islamabad for like 3 or 4 times to organize meetings and bring others at the technical level to have discussions, but then at the government level to facilitate other kinds of discussions. Also had other engagements and had the opportunity to meet other world leaders. This is a picture with the Pope. I was invited by the Pontifical Academy of Sciences to discuss the potential role and implications in church theology for emerging and converging technologies. And here's a picture on the right-hand side with the late Fidel Castro Jr. Fidelito was early on involved in the normalization of relations between the US and Cuba. And my office and I played a small role in the normalization and signaling that the Cuban government was open to the normalization of relationship between the two countries. Of course, that was during the Obama years. 2 weeks ago, I was driving and crazy because I was not very responsive. I was on travel. This is a picture of where I was having conversations. That red line is the border between Poland and Belarus. I took a picture, a screenshot on my Google Maps. That's the location where we were having conversations with officials from the Belarusian government and the Polish government, as we are putting together science diplomacy effort to bring them together to coordinate on the genetic variability and preservation of genetic resources that are unique to that forest in the border between Poland and Belarus. We are currently planning a meeting there next year. Hopefully the political situation will be easier than this time around, but this is part of the science diplomacy angle that we do here at the Academy of Sciences. And currently, I am deeply involved, and you might have heard my phone is, even though I have 3 phones, so I forgot to mute one. And we're helping families and scholars in particular from Afghanistan that had a relationship with the government, and some of those programs we were implementing from the academies. Here's a picture of one of them that we were able to help them navigate Taliban checkpoints about 3 weeks ago. They're currently in Doha. The picture on the left, that's the family reunited in Doha as they wait for the US government to process their paperwork and vetting process to bring them to the US. We still have 19 individuals there of 5 families that we are still trying to find ways to securely provide safe transit. We engaged the president of Rwanda, who personally agreed to host some of those families, and I am ready to go to Rwanda to meet the families and facilitate their safe transit. And that should happen, if everything goes well, in 2 weeks. Other things that I do at the academies, this is another meeting that I organized from the scientific framework to putting together the meeting. It was the Nobel Prize Summit that we did this past April. You can see the link, that everything is still live if you want to see. But I want to end with this video that Laurie Garrett, Jennifer Doudna, and Peter Doherty, in a conversation they had with Dr. Anthony Fauci.
Tony Fauci, I want to come back to you because as soon as we start talking about climate change, we are, whether we like it or not, we're talking about politics. And I can't think of any scientific figure, maybe since Galileo and Copernicus, that have dealt as deeply and effectively, in your case, with that interface between science and politics. You know, I mean, I remember back when you were trying to convince Ronald Reagan that he had to take HIV/AIDS seriously. And when you were fighting, you know, every kind of battle related to Zika, to one disease after another, with every single president since 1984, both parties, and with some pretty tough cookies on Capitol Hill. And you've managed to protect budgets, you've managed to protect science as a mission, and now you find yourself in this huge lockdown battle, as you described, in the most deeply divided America. Many say that we have seen since our Civil War of the mid-1800s. What are the lessons here? And in particular, there aren't very many scientists who have your skill set in terms of politics and the ability to speak to average citizens or less than average politicians. Where do we go from here? How do we develop that skill set? How do you transfer your skill set? And how do we protect the mission of science as it goes into ever more controversial territory?
Well, Lori, I think the most important thing is to just maintain your integrity of sticking with the science. And sometimes the science leads to inconvenient truths that make people very uncomfortable. And you just have to make up your mind that the most important thing to do is to maintain your own integrity and the integrity of the science. You ask about dealing with different presidents of different persuasions. The first time I went in to see Ronald Reagan and to deliver the inconvenient truth that I thought he should have been more using the bully pulpit of the presidency to alert the world that we were dealing with an emerging catastrophe. When I went in there, a good friend, an old person who had experience in the White House, told me something that I've lived by for the last 37 years of advising 7 presidents. And it was a very interesting, simple thing he told me. He says, when you go into the White House, every time, and you walk into that, open the door of the West Wing, tell yourself this may be the last time I'm ever going to go in there, because I might have to tell the president something he doesn't like, and I might not get asked back. When you go in saying, wow, I'm awestruck, this is a really amazing place, I want to get asked back. Once you fall into that trap, you may compromise your integrity and start telling people happy talk when really you need to tell them the truth. And what happened is that I was fortunate because I dealt with presidents who were able to see that, respect me, and ask me back. So, if I had gone in and given happy talk all the time and didn't stick by the science, I think likely they would have lost respect for me and they wouldn't have asked me back. So the lesson is speak the truth and just stick with the science.
Speak the truth, stick with the science, don't deliver happy talk. That's a pretty good way to wrap it up. We have reached our time limit. Tony Fauci, I want to come back to you because.
So that's it. I'm going to let Lori finish the talk for me. I'm happy to answer any questions. Thank you very much. Thank you for your attention.
Thank you. That was truly fascinating. Are there any questions?
I have a question. This is really fascinating talk, and thank you very much. My question is, as a PI, I would like to have my graduate students to be exposed to many different kinds of job opportunities. So how would you suggest to the young budding graduate students who are interested in science policy, what would be the path that could prepare them to go into science policy?
So there are different paths, and it really depends on where they are. The truth is that you have to meet people wherever they are and wherever they want to go. So it could start by simply volunteering with a professional society. It could be simply by joining a local student chapter that may be doing some volunteering work as it relates to policy. You don't have to join government to help inform policy. That's the advocacy part. So there are different ways they can volunteer in their local community with different government officials. Most have local congressional offices. So there are different ways that they can engage. You can start taking a class to understand how government works and the different components of government. Depending also on their interest, languages become very important. Social science classes to have a deeper understanding of culture if they have an interest in becoming a regional expert of sorts. So it really depends where they are and where they want to go. There are different paths. But for a PI, I would say, sorry, it's just to keep that open mind that not all students have to end up being like the professor. There are different paths, there are different ways, and simply as we know, academia does not have enough room for all of the students that we're training. So professors need to have an open mind and allow those students to explore other options. But that doesn't mean for the students that they should neglect their research. They're there to complete their degree. They're there to comply with whatever fellowship conditions they have and all of those things. So it's a balancing act.
I think everyone is humbled into quietude. Are there any other questions?
So I have a question. You mentioned that there was a civil unrest which stopped you from doing the research. So I was wondering, what are a lot of the barriers that people face that aren't able to get into science, or they don't have the opportunity to get into science, especially in like developing countries? And do you think there's any ways that we could overcome these barriers and get possibly more science within these communities?
That's a very complex question, and one that does not necessarily have an easy answer. There's, for example, the challenge. I'll use the example that we're dealing with in Afghanistan. That's what the government was trying to do there for the past 20 years, provide scientific collaboration, provide funding, develop a group of scientists, a community there that could help Afghanistan stand on its own in terms of science. However, currently with the new Taliban regime, that affiliation with the US is the one thing that's putting these scientists at risk, because they were collaborating with the US. That's why we're involved in trying to provide safe passage and help them. It starts with the local government. It starts with a desire by local leaders to encourage free thinking, encourage what we take for granted here, the scientific discourse, the ability that you have to question your other students, you question your professors. That's something that we learn, something fundamental when we're doing our degrees here. That's not necessarily true in other places. It starts there. When you have conflict, bias, rules, regulations that prevent you, and it's frowned upon by institutions to do certain things, that's also something that needs to be in place. A fair process for peer review, for awarding funding, all of those things are elements that have to be in place for scientific community to develop, not only in developing nations, but also to remain strong in developed countries. Sometimes we are quick to point the fingers to developing countries, but developed countries also have a responsibility to do that and ensure that their scientific community are not put aside or neglected or asked to do and say certain things because they don't have that flexibility. I served over a year and a half with the Trump administration. Even though I faced pressure for them, I decided, and I told them many times, that I was a scientist. And my job was to solve the science variable. I was not going to say one thing that would favor one or the other. I asked them to do that with the regional experts. The science says something, and if it was China and they needed something, then they should go talk to the Chinese desk. Or North Korea, I had some engagements with early on with the group that was helping and trying to get the US closer to Kim Jong-un. And it was the same thing. So it's not only true for developing countries, but also for developed countries. We need to ensure that the scientific community have a way to conduct their science without interference.
Okay.
Franklin, I agree. I mean, this is fascinating insight. I guess I have two comments. The first one is, what happens to these Afghani scientists or engineers when they get taken out of the country in a situation like this? Are they ever able to, develop their scientific careers in other countries?
So that's what we're doing. So early in this process, the president of the Academies wrote a letter to the president of Rwanda, I'm sorry. And he personally approved and agreed to host them. The scientists that we supported in Afghanistan are water experts, and Rwanda has a water problem. Also, I think we do have that problem here in the US, in some parts of the country. So having this expertise and creating meaningful opportunities for them to remain connected to their professional identities is one of those things that we are looking for. We're engaging the White House, the State Department, USAID, and other relevant agencies to make sure that these refugees end up, or evacuees at this point, end up in positions that relates to their professional identities, that they don't end up driving an Uber around DC because they're not able to find a job relevant to their scientific expertise. So that's something that we're putting together. I oversee a group here at the Academies that does human rights. And one of the lessons learned from other refugee experiences in the past is that we bring them here or relocate them to other places, and without regards to expertise that they may have, which makes it harder and more difficult for them to become productive members of society. So we're piloting something different, and hopefully it will work.
Yeah, what a difference you're making. I mean, I know those are difficult negotiations, but it's fascinating to get the inside look at that. I should mention, not to you so much as to the trainees, I've had two students, and I'm sure there are others here, who've gone into science policy through a more traditional route. There are fellowships through the AAAS. And then those people, one of my students is with the Veterans Administration, and one of them was in the director's office at the NIH on a fellowship. And she then she said, I could go up in this kind of administration, but I really like helping scientists. So she's director of one of the programs in Payne, actually. But there are lots of ways to get into science policy, I think, is what I'm trying to say. Lots of different levels. Not all of them are as impactful as what you're doing, but it's really, really interesting to hear what you've been up to. Thank you.
Thank you. Thank you for the invitation.